In the 1600s, science had no knowledge of what life looked like on the smallest scale. People wondered, what are plants and animals made of? How do they grow? The answers began with a simple piece of cork and a curious mind. In 1665, English scientist Robert Hooke placed a thin slice of cork under his microscope. What he saw astonished him, a honeycomb of tiny box-like compartments. He called them cells, after the small rooms, or cellula, in monasteries. But what Hooke saw were only the empty cell walls of dead plant tissue. The true living part of the cell, its internal machinery, remained invisible to him. A few years later, in Holland, Antony van Leeuwenhoek built simple but powerful microscopes with lenses he ground by hand. Looking at drops of pond water, scrapings from his teeth, and even human blood, he discovered an entirely new world of living cells. He saw bacteria, protozoa, red blood cells, muscle fibers, and sperm cells. He called them animalcules, little animals. Hooke had named the cell. Van Leeuwenhoek revealed that cells were alive. For the next 2 centuries, scientists debated a fundamental question: how do new cells arise? Some believed in spontaneous generation, that life could appear from nothing. Others suspected that cells must come from pre-existing cells, but no one could prove how. By the early 1800s, microscopes improved dramatically. Scientists began using stains to color cells, making hidden structures visible. One of the first was carmine, a natural red dye from insects. Later, the invention of aniline dyes, synthetic chemical colors like mauvine, gentian violet, and fuchsine, lit up cells with striking contrast. For the first time, the nucleus of the cell could be seen clearly, and inside the nucleus, something strange appeared during cell division. In 1840, German scientist Robert Hooke studied chick embryos under the microscope. He saw nuclei splitting into 2 and concluded that new cells come only from the division of existing ones. A few years later, Rudolf Virchow popularized this idea with the famous phrase, omnis cellula, a cellula, every cell from a cell. This was a breakthrough, but the exact steps of how a nucleus divided were still a mystery. In 1842, Swiss botanist Carl Wilhelm von Nageli made a key observation. Studying the hairs of the spiderwort plant, Tradescantia, he saw delicate thread-like structures appear inside the nucleus during cell division. He called them transitory cytoblasts. Today, we know them as chromosomes, the carriers of genetic information. Though Nageli didn't realize their importance, this was the first recorded sighting of chromosomes. Over the next decades, others continued the work. Eduard Strasburger in plants and Otto Büchli in animals described similar nuclear threads, hinting that division followed a precise pattern. Still, no one had mapped the process step by step. That achievement came in 1879 thanks to German scientist Walther Flemming. Using aniline dyes on salamander embryo cells, he observed the tiny nuclear threads moving in beautiful patterns. He saw chromatin, the substance inside the nucleus, condense into visible filaments, line up in the center, split apart, and move to opposite ends of the cell. Flemming named this process mitosis, from the Greek word mitos, meaning thread. For the first time, the mystery of cell division was revealed as an orderly sequence, not a disorderly event. In 1888, German anatomist Heinrich Waldeyer gave these threads their modern name, chromosomes, color bodies, because they stained so vividly with dyes. The term captured both their striking appearance and their central role in the life of the cell. But what exactly were chromosomes for? Were they just structures of cell division, or something more? The answer came from the brilliant German biologist Theodore Boveri at the start of the 20th century. Boveri studied the fertilized eggs of sea urchins, an animal perfect for observing cell division. By carefully manipulating these eggs, sometimes allowing them to be fertilized by more than one sperm, he created embryos with the wrong number of chromosomes. The results were striking. Embryos with the full balanced set of chromosomes develop normally. Embryos missing chromosomes or with extra copies grew abnormally and often failed to survive. From this, Boveri concluded something revolutionary. Each chromosome carries unique and essential information. The complete set of chromosomes is required for normal development. In other words, chromosomes were not just colored threads. They were the carriers of heredity, the physical vehicles of genetic instructions. This idea laid the foundation for the chromosome theory of inheritance, later confirmed by genetics. The story of mitosis is the story of science itself, of lenses ground by hand, of brilliant stains, of careful drawings, and of centuries of questions. From Hooke's empty cork cells, to van Leeuwenhoek's animalcules, to Naegeli's mysterious threads, Slemming's beautiful sequence of mitosis, and Boveri's sea urchins, each discovery built upon the last. Today, every biology student can recite the stages of mitosis: prophase, metaphase, anaphase, telophase. But behind those textbook diagrams lies a centuries-long journey of curiosity and persistence. Mitosis is not just a cell dividing, it is the engine of growth, repair, and life itself. And its discovery shows us how patient observation and simple tools can uncover the deepest secrets of nature.